Hi, my name is Julie Krämer and I'm PhD student at Forschungszentrum Jülich. In this video I give a short insight into my research, which is the collaboration between Shoot Group at Forschungszentrum Jülich and Agroecology Group at Bonn University. Can we achieve a sustainable intensification in agriculture by introducing crop mixtures?
In modern farming, arable crops are usually uniformly grown in large fields referred to as monocropping. Here, the same plants are grown side by side to allow efficient and easy crop management. But what would happen if we placed very different plants in between? As shown here, an ancient but nowadays rarely used diversification strategy is the simultaneous cultivation of legume and cereal plants, namely mixed or intercropping. Benefits of mixed cropping are improved root growth and high soil fertility. This is due to a complementary soil resource exploitation. The nitrogen fixation of legume plants allows a very efficient nitrogen use. Other nutrients as well as water are better accessible due to improved root architecture. Moving above ground, an additional benefit can be seen in the heterogeneous canopy architecture. Light interception is very high in mixed stands. And lastly, legume plants attract a lot of pollinators, which are beneficial for the agrobiodiversity. However, designing mixtures is a difficult task because genotypes can behave differently when grown in a mixture compared to the sole cropping system. Thus, mixtures do not always outperform monocultures. In my PhD project, I will therefore investigate the physiology of different mixtures, combining genotypes with contrasting structural traits such as plant height. In a field experiment in 2021, we have grown mixtures of faba bean and spring wheat and their respective sole crops. We have tested 3 different wheat varieties and 2 different faba bean varieties with contrasting structures. Combining those resulted in 6 different mixtures. In this diagram, we find grain yield data of the 11 treatments. Yield levels of sole cropping plots of faba bean are shown on the left, spring wheat yield is shown on the right, and the mixture's yields can be found in the center of the plot. To get a better proxy for the productivity of the mixtures, I would like to introduce the concept of the land equivalent ratio. Here we first divide the cereal yield of the intercrop or the mixture by the cereal yield of the monoculture. Next, we divide the legume yield of the mixture by the legume yield of the monoculture. The sum of both gives us the land equivalent ratio and tells how much monocrop land is needed to harvest the same amounts as grown in the mixture. If the LER is larger than 1, the mixture shows high performance. If the LER is below 1, the mixture performance is bad. In the field experiment, we found ratios of at least 1 for 4 of the mixtures. It was significantly higher than 1 for only 2 mixtures. So we might ask, what are the drivers of mixture performance? I already mentioned the increased light absorption, complementary nitrogen use, and an overall efficient resource acquisition. But what do those factors have in common? They drive the most basic principle underlying the performance of all crop plants, which is photosynthesis. Let's have a closer look at this and see what is happening when light is entering a crop canopy. Not all sunlight is absorbed or transmitted through the canopy. Some light is directly reflected by the leaves and not used for photosynthesis. However, large amounts of light are entering the leaf cell and arrive at the chloroplast. Here different pathways may happen for a photon. Some of the photons are hitting the photosynthetic machinery and drive photosynthesis, which is the most important process that drives biomass production and finally yields. To protect the plant cells, some light is emitted as heat. Other photons are not used at all and are emitted with a lower energy level. This emission is called fluorescence. It is related to the photosynthetic efficiency of the leaf and can be detected by high-performance sensors from ground and from air. In this photo, you see me with a point spectrometer, which makes it possible to passively measure top-of-canopy fluorescence. Small fiber optics are not only collecting fluorescence but also reflectance signals. The latter makes it possible to calculate a proxy for photosynthetically active radiation, which is absorbed by the canopy, shortly APAR. Here you see plots of APAR against the total fluorescence signal of a canopy on a sunny day in June. We can observe the high levels of absorbed radiation in the mixture plots on the left side. Also, we see a positive relationship between APAR and solar-induced fluorescence. This means the more light is absorbed by the canopy, the higher becomes fluorescence. To get a deeper insight into the variation of fluorescence emission, total SIF levels are shown for each treatment. Now we see significant differences between the two monocultures, but no significant differences between the mixtures. However, the dataset is rather small and SIF signals are very sensitive to the illumination conditions during the measurement period.
Next step in my PhD project is to study SIF signals from air, which allow a higher spatial and temporal resolution and larger sample sizes. We believe that using non-invasive sensing technologies is an innovative approach to study diversified cropping systems. It facilitates new insights into the complex relationship underlying the mixture of two species from the leaf to the field level. Thank you for watching this video, and I'm looking forward to listening to your questions and for further discussions.